In this video, we'll be talking about the polymerase chain reaction or the PCR. It is one of the most important step in the recombinant DNA technology. Now, the PCR actually means the amplification of the gene or DNA segments. That is, it is the amplification of the gene or DNA segment. In other terms, if we say, it is the process of making multiple copies of the gene. So it is amplification of genes, that is the making multiple copies of that particular gene. And selective amplification is carried out by a process which is known as polymerase chain reaction. Now, why we are using the term polymerase chain reaction? Because polymerase enzyme is required for this chain reaction to amplify or to make multiple copies of this particular gene. And this method was discovered by Karamolis in 1985. So Karamolis was the person who discovered this in 1985, and it is one of the important steps of the recombinant technology. Now, over here, this is basically divided into three again major steps. It is further divided into three major steps. Now, when we are talking of the principle, let us talk about what is the principle behind this. The basic principle of this method or technique is that when a double-stranded DNA molecule is double-stranded, so when this double-stranded molecule of DNA is heated at high temperature, the two strands of the DNA molecule will separate due to the breaking up of hydrogen bonds, and as a result we will be having two single-stranded DNA molecules. Now, so if we use these two single DNA stranded molecule as templates and then can make a copy with the copy of this, which will be a complementary copy obviously with the help of an enzyme polymerase, then multiple copies of the original DNA will be formed. And if the process is repeated a number of times, multiple copies of the DNA segment can be generated. So what we are doing, if in simple terms, if we understand, it is the DNA molecule is having double-stranded structure. If the hydrogen bonds separate are made to separate, then we have two single strands, and if we use the polymerase enzyme, we can synthesize or produce exactly the same copy, multiple copies. And if these two are further separated, again we can have multiple copies obtained by this method. So polymerase reaction is an important step again, and it is done in the presence of polymerase enzyme. Let us now study what are the actual parts or what is the actual process. As I told you, the process is basically divided into three major steps, and the first step in the PCR technique is denaturation. The first step is denaturation. The second step is known as annealing. And the third step, which is here, is known as extension. So these three are the major steps which are the part of the PCR technique. So these three are the major steps. Let us study one by one each of the step in detail. Now first is, let us talk about the denaturation process. denaturation, that is over here the desired, we call it as a template DNA. So the desired DNA fragment, that is, it is heated, which we call it as a template over here. So this will be known as template. It is heated to a temperature that is 92 to 94 degree Celsius. 92 degree Celsius to 94 degree Celsius. Heating is done. Now, this heating actually results in the separation of two strands of DNA. So if the two strands of DNA, one is 5' to 3' and the other will be 3' to 5'. Over here, they are held by the hydrogen bonds. Now, due to the heating, these hydrogen bonds will break and it will result into the separation of the two strands separately, like single stranded. Now over here, each of the strand will function as a template. So over here, this is one template and this is another template. So we have two templates for the synthesis of new molecules of DNA. Now over here, it is very important to maintain the temperature. The temperature needs to be maintained. The reason being, the temperature should be maintained long enough. This 92 to 94 degrees Celsius should be maintained for long enough so as to allow the DNA strands to break the hydrogen bond completely. But if the temperature is more, or if it is for a very long duration, it may result in the breaking or the loss of other molecules as well, or there may be some not proper breaking up of the hydrogen bonding. So therefore the temperature needs to be maintained, and this process of denaturation normally takes 15 to 30 seconds. So this is a very short duration process. In the denaturation, the desired DNA fragment is subjected to a high temperature. The temperature ranges 92 to 94 degrees Celsius, where due to the heating, the hydrogen bonds will be broken up, and as a result, we have now two single-stranded DNA which will act as a template, and the correct temperature for a certain period of time is important so as to allow the hydrogen bonding breaking. Also, the approximate time for the process of denaturation is around 15 to 30 seconds. The second step, now the first step, after the first step, the second step is annealing. Annealing, it is a longest and it is very important phase. Annealing process, it is the process in which the two primers, that is we have created, hybridize to each of the strands of DNA. Now this step is carried out a lower temperature. So over here the temperature is low, and low temperature, that means around 50 to 65 degrees Celsius. So it is conducted at temperature, low temperature, that is we talk of 50 degrees Celsius to 65 degrees Celsius. And it actually, the temperature, we are putting up this range because this depends, the temperature depends on the length and the sequence of the nucleotide primers. Now what happens is, why do we need to lower the temperature? Actually, when we lower the temperature, it allows the primers to attach to the specific locations on single-stranded template DNA. So what is happening over here? The two strands which are being produced by the denaturation process, the low temperature will allow the primers to attach to these single-stranded DNA, that is the template DNA, and this binding is normally seen at the 3' end of the strand. So suppose if this is 5' to 3' and this is 3 dash to 5 dash, the binding of the primers will be taking place towards the 3 dash end of the DNA template. Now, when this primers are actually single strands of RNA and DNA, and they are normally 20 to 30 base pairs long. Like, they are short fragments, maybe of RNA or maybe of DNA, but the primers have a base pair, primers around 20 to 30 base pairs. So these are attached at 3 dash end. Now, the temperature maintenance is again very important criteria during the process of annealing. Now, what happens is, if the temperature goes high, the primers will be unable to bind to the template. So, if the temperature is high, if high temperature, then the primers will not be able to bind with the template DNA. And second is, if the temperature is low, very low, if the temperature is very low, the DNA will not get completely denatured and the amplification rate will be very low. So for here, we need to have a proper maintenance of the temperature. Now, the primers are very important over here because they are the starting point for the synthesis of DNA. When we are talking of the annealing process, it is attachment of the primers to the single-stranded DNA molecule, whereupon now the polymerase enzyme can act, because the polymerase enzyme can only add DNA bases to a double-stranded DNA. It cannot start on its own over here. First, the primers have to be there, and the synthesis will then further take place. Next is after the attachment, the two separated DNA strands are complementary and they run in opposite directions, that is 5 dash to 3 dash and 3 dash to 5 dash. Therefore, there are two primers, a forward primer and a reverse primer. So in one of the, we can say one of this is, because this will grow in this direction and this will grow in this direction. So this will be a former, and this other one will be a reverse primer. This will be a reverse primer and this will be a forward primer. In this direction and in this direction. Now, this step of annealing usually takes about 10 to 30 seconds. So, the entire process of annealing requires around 10 to 30 seconds. Again, it will depend on the length. It will depend upon the length of the DNA fragment. The last step is the extension. That will be the third step, that is the extension step. Now, extension is again the process in which the Tuck polymerase, there is an important enzyme. Third, we are talking of extension. Now, extension, in this process, the Tuck polymerase synthesizes DNA in the region between the two primers. So over here, we are talking of a special type of enzyme, which is Tuck polymerase. And this Tuck polymerase over here is used to synthesize DNA in the region between the primers by using DNTPs and magnesium ions, etc. Now over here, actually it is the process of addition of the nucleotide bases. Now over here, the temperature which is required is 72 degrees Celsius. 72 degrees Celsius is the optimum temperature. Now what happens is, this Tuck polymerase will attach to the DNA primer and will add DNA bases one by one in 5 dash to 3 dash direction. Always remember, the polymerase enzyme will always act in 5 dash to 3 dash direction. Also, it will attach to the primer it will attach to the primer and will synthesize base, nitrogenous bases. So as a result, the length will be increased. That is why it is known as elongation or extension. Now the cycle is repeated, that means multiple times it can be conducted according to the need. If the process of replication is repeated several times, the segment of DNA is amplified approximately billion times, and then the amplified fragment can be then ligated with a vector for further cloning. Now the duration of this particular step, that is extension, depends upon the length of the DNA sequence which needs to be amplified. But normally the extension takes place approximately 1 minute. So this is approximately for 1 minute, and but it further depends, and 1 minute to copy approximate 1000 DNA bases. So within 1 minute you can make 1000 DNA bases. So depending upon the length of the DNA fragment, we can have different timings, and the temperature required is 72 degrees Celsius. This completes the PCR reaction.